Welcome to my video about ubiquitin and just enzymes in general. This is for Investigation 13, Part B, Enzyme Research, and it will be referring to ubiquitin, which is an enzyme, and a little bit about enzymes in general. Before we jump into ubiquitin itself, I thought that we should review some key features about enzymes. The first being what an enzyme is. An enzyme is just a chemical that speeds up reactions. There are proteins that have an active site, which is where the reaction happens, where the substrate fits into this little place, and it also lowers the activation energy, which makes it easier for the chemical reactions to kind of break apart, which is why the things are going faster when an enzyme is present. They must fit perfectly. There's two theories to this. The first being lock and key, which means that it fits perfectly into it, like we would fit into a lock, and the other one being induced fit, which means that the active site changes to fit the substrate. Another thing that's good to know is that they need to be activated. This can happen by cofactors, which are inorganic molecules, which means that they do not have carbon, or by coenzymes, which means that they do have carbon because they're organic molecules. They can also be turned off through competition inhibition and allocentric inhibition. Both of those things pretty much just cover the enzyme not allowing certain to happen. The first one being because it actually blocks it. The second one being a blockade, like the side, stopping the reaction from happening. There are certain factors that will affect an enzyme. The first thing is pH. If the pH is too high or too low, then the enzyme just denatures, meaning that it breaks down, so the reaction cannot occur. Same thing with temperature, and then also substrate and concentration affected, because the more substrate or concentration of the solution that there is inside of the thing is obviously the faster that it will go. So now that we know a little bit about enzymes, we can talk about ubiquitin. So kind of like the word ubiquitous, it's found in many places. So some of the things that ubiquitin does do is that it's conjunctuated to cyclin during the G1 phase of mitosis. This means that it plays a really important role in controlling the cell cycle itself. It also has to do with DNA repair, abiogenesis, controlling transcription, and cell death. We've talked about the structure of ubiquitin. It's just seeing what ubiquitin is. So here to the left, you see the two most common states of ubiquitin. Like I mentioned earlier, since it is so important to us, we're not going to find a lot of different types of ubiquitin because you can't change the structure without completely degrading and the whole system of a cell, because it does control things like the cell cycle. It's fun actually, since yeast to us, we've only actually found 3 sequence differences that everybody shares with ubiquitin, and that is due to its importance. So how does it compare from organism to organism? Well, I just kind of touched up on that. Since it has evolved to be since it's involved in such an important process, its structure does not change much from organism to organism. If you change the structure of it, you change the function, and you can't change the function of the cell cycle without it completely being detrimental to the organism. Another question here is, does this mean the enzymes evolve numerous times? Well, as I explained before, since it is so important, it did not evolve numerous of times because everybody has essentially the same cell cycle. Everybody has essentially the same things that ubiquitin is involved with, hence why it's ubiquitous and hence why it is so important and why it is the same. So how does this affect the similarity of the DNA or this enzyme? Well, for the reason that I already explained, the similarity across everybody is pretty much the same. The only thing is that you don't find it in eukaryotic cells and in archaeal bacteria. How do organisms survive in extreme environments like hot springs? Well, organisms themselves that do involve in these extreme environments, they're called extremophiles. These extremophiles can be found in hot springs, like the question mentioned. They can be found in this, like volcanic pits, places that most organisms, or we presume, cannot survive because the environment is so harsh. Now, organisms in general survive in these places just by adapting to them. So usually you're not going to find a human living in a hot spring. But you could find some microorganisms that do because they've adapted to that type of lifestyle. They somehow get energy through the energy that is provided in that harsh place, and they make it work. So for my enzyme in specific, this ubiquitin enzyme, it's not found in archaeobacteria, which is what is found in a hot spring, so it does not survive in these really extreme areas, and it's not like it helps an organism survive in really extreme areas, because as we described earlier, these are enzymes, and enzymes are proteins, and proteins denature, and one of the factors that do affect that is temperature. So if this ubiquitin did live in a hot spring, it would completely denature. And that concludes this video. All the resources that I use will be in the description down below. Thank you to homepages.bw.edu, which is where I got pictures from and where I got a lot of this information from, and also my science teacher, Mr. Lima, which is where I got most of these questions derived from, and there are the answers. So I hope this video helped you, and I hope you enjoyed it, and thank you for watching.